friends. Today we are going to be talking about cell division, and you need this handout in order to take your notes. So we're going to zoom in on this first box, and that is where we are going to title this. So today is a day about cell division. And cell division, in this case, is when one cell divides into two and the two cells are identical. They're identical to themselves and they're identical to the original cell. So that's what I have here. I've got one cell and it divides into two. Each of those then divide into two as well. And this process is also called mitosis. So let's zoom in on this process. So what I have here is I have a cell, just a regular ordinary cell. Inside of the cell is a nucleus. And inside of the nucleus are the unreplicated DNA. And what that means is that each of these chromosomes of DNA is just a single. So you've got this one here, and that would be like your chromosome from mom. And then you've got another one here, and that would be like your chromosome from dad, and they would have the same genes on them, but you only have one copy of the gene from mom and one copy of the gene from dad. So in this cell, we have 3 pairs of chromosomes. And those chromosomes are found in the nucleus. They have one copy of each chromosome from each parent. So we have a pair of chromosomes here. This is what we mean when we say a pair. There are 2 that are the same length. The centromere is in the same place, and they have the same genes. You've got one of these is from mom, and one of them is from the dad. And then this would be an example of a single chromosome. And chromosomes are the way that DNA is stored. So chromosomes are really just long strands of DNA. And then genes are segments of DNA that code for specific traits. So, for example, I have one gene here, so we could say that this is gene 1, and it's going to code for a specific trait, and then this is going to be gene 2, and this is going to be gene 3. And not all of the DNA in a chromosome is actually genes. Not all of it codes for proteins and traits. So they're really just these gene sections or segments, and that's going to code for a trait. And then this other part of the DNA, sometimes we don't know what it is or what it codes for, and sometimes it's just junk DNA. It's just there. So when we said over here that we have unreplicated DNA, this is an unreplicated chromosome. Whereas here we have a replicated chromosome. And when a chromosome replicates, you end up with this traditional X shape. And what you have is you have identical chromosomes. So you've got one version here with genes 1, 2, and 3, and then the same exact chromosome here, replicated perfectly, genes 1, 2, and 3. And gene 1, whether it's on this one or on this one, it's going to code for the exact same trait because it's the same version of the gene. This is gene 1, and it's identical to this gene 1. The first thing that happens in mitosis, the first stage of the cell cycle, is called interphase. And in interphase, there are actually 3 different stages that occur. The first stage of interphase is called G1. And in G1, the cell grows. Next, you have the S stage. And in the S stage, DNA is replicated. So we're going to go from one chromosome to this X replicated chromosome. And then in G2, the last part of interphase, the cell is going to prepare to divide. And so what we have here is we have the replicated DNA still inside of the nucleus. So if we compare this image to this image, we can see that each of these single strands of DNA has now been replicated and it has its perfect match on top of it. Once the cell is ready to divide, it's going to go into the next phase of mitosis, which is prophase. And in prophase, the DNA condenses into chromosomes. So what we saw in this previous picture here, this is replicated DNA, but it's just kind of loose. It's all tangled up in the cell. And so in order to divide, it needs to organize this DNA so that it's able to separate the DNA into the different cells. And so when it condenses and it forms these chromosomes, this is all coiled up neatly. It's neatly organized so that it can then divide. So what we have here is we still have the same 3 pairs of chromosomes. So we have, for example, this chromosome right here, which could be a chromosome that you got from your mom, and it is paired up with a chromosome that you got from your dad. And this is a pair of chromosomes. So we have 3 pairs. This is one pair, this is another pair, this is another pair. We've got 3 pairs for a total of 6 chromosomes in this cell. Now, we say that there are 6 chromosomes in this cell, even though the DNA is replicated. And that's because we're actually counting by these centromeres. And when we look in a microscope, we can actually only see kind of an individual centromere like this. And so we would count 1, 2, 3, 4, 5, 6 chromosomes, even though each of these has 2 copies of that chromosome. All right, after prophase, we get metaphase. And in metaphase, the chromosomes line up in the center of the cell. So here, we still have our pairs of chromosomes. We've got, this is a pair. And we've got this one, which came from mom. And we've got this one, which came from dad. And then we've got another pair. This one is actually a pair with this one right here. Their centromeres are in the same place, and they have the same length and size. And then this is another pair. So they don't necessarily line up next to their partner, but they all line up in a row. The next phase is anaphase. And in anaphase, the sister chromatids are pulled to opposite sides of the cell. So this here is one sister chromatid, and this here is another sister chromatid. So this is one of those sister chromatids, and this is the other sister chromatid. And so what you're going to end up with here is you're going to end up with this sort of divide that's happening. And the divide is going to make it so that on this side of the cell, you have one of those big copies, the biggest chromosome that we had from mom, and on the other side, you've got the other copy of that big chromosome from mom. So you're going to end up with 6 chromosomes, one of each chromosome that you had before, exact same, and then here you're going to have another 6 chromosomes, again, one of each that you had before. All right, the last part of mitosis is called telophase. And in telophase, the cell splits into our 2 identical cells. So we've got one cell here and one cell here, and they have the same number of chromosomes. They have the same versions of chromosomes, and they've got 2 copies, one from mom and one from dad, of each of those chromosomes that make up a pair. So on our original chromosome, we had genes 1, 2, and 3. And so here, we're still going to have gene 1, and then gene 2 and gene 3. And then this chromosome in this cell, it's identical, so it's also going to have gene 1 and then gene 2 and gene 3. Now, in order for this to take place, something called cytokinesis has to occur. And the word cytokinesis gives us some tips about what is happening. So cyto refers to the cytoplasm, and kinesis is splitting or moving. And so cytokinesis is when the cytoplasm divides between the two cells. And so inside of these cells, you have all of your normal cell parts. So you've got some ribosomes, you've got your cytoplasm, you've got some mitochondria, and those things are just going to get divided between these two cells. But we say that these cells are identical because their DNA is identical. Their DNA is identical, which means it has the same genes and it has the same versions of those genes. So those cells are going to have the same traits. And that, the whole thing put together, is how a cell divides through the process of mitosis.